Hello, everyone. Welcome to today's TSU Technique Talk. Technique Talks, a series of educational courses, will answer the question, how can I get this experiment to work? Course instructors will teach about how to get a new method or technology set up in the lab, common challenges or pitfalls to avoid, and troubleshooting steps that will help scientists achieve publication-worthy results. I'm Nikki Spahich, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our instructor, Dr. Laura Pellegrini, will discuss the critical factors that ensure great 2D stem cell culture. I'd like to take a moment to thank our webinar sponsors, Sinobiological, the Baker Company, Biotechne, and Millipore Sigma. Our sponsors have provided us with some helpful resources related to today's course, and you can download them directly from the Handouts tab, located on the right side of your screen. This course is meant to be interactive. Please send us your questions or comments at any point during the discussion, and the instructor will address as many as possible during the live Q&A. The Technique Talk will be archived on the Scientist website. We will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. And with that, let me introduce our speaker, Dr. Laura Pellegrini, a postdoctoral research fellow at the MRC Laboratory of Molecular Biology in Cambridge. She studied biomedical sciences at the University of Padua, Italy, where she worked with Elisa Greggio, who inspired her to pursue a career in neuroscience. Pellegrini did her master's degree research on neuropathic pain at the University of Paris. She then joined an international PhD program shared between the National Institutes of Health and University College London in the laboratories of Kirsten Harvey and Mark Cookson. She studied the genetics of Parkinson's disease using both cellular and animal models and completed her degree in neurogenetics in 2017. Pellegrini now studies cerebral organoids in the laboratory of Madeline Lancaster at the University of Cambridge, where she recently developed a model to study the choroid plexus and cerebrospinal fluid secretion in vitro. All right, let's make sure your slides are running. Okay, Dr. Pellegrini, take it away.
Okay, thank you very much for the introduction and good afternoon, everyone. Thanks for joining this seminar. So today I'm hoping to give you an overview of the technical aspects of working with stem cells. So this is my talk's overview. So I'll give you a brief introduction on what are stem cells and how we can distinguish adult and embryonic, iPSC and ESCs. How do we recognize stem cell colonies and the quality controls that we need to do? How do we maintain stem cells in culture, the feeder-dependent versus feeder-free methods, and I'll give you at the end an example of application of stem cells. So stem cells are essentially the body's raw material, and they basically are cells that can self-renew and progressively differentiate in specialized cell types. So they exist both in embryos and adults. And in this diagram, you can see actually stem cell here that they can give rise to different type of cells here, nerve cells, cardiac cells, and liver cells. And here you can see even the cardiac cells, they can be contractile. So why are they so interesting and what can they be useful for? So there's multiple applications. They can be used to increase our understanding of diseases, and by watching stem cell to basically mature over time in different tissue, we can understand better how conditions develop. We can generate healthy cells to replace disease cells, and this is an application of used in regenerative medicine, for examples, and they can be used to test drugs for safety and effectiveness. So the concept of cell potency is that basically this is a continuum and it reduces each step of specialization during development. So during development, we have an initial state of totipotency, which is basically the zygote. And then we have a separation at the blastula stage or the blastocyst stage in two different lineages. So the external lineage here is called the trophoblast, which gives rise to the extraembryonic tissue, such as the placenta. And then we have this inner cell mass of the blastocyst, which can be extracted to then generate embryonic stem cells. And these embryonic cells are pluripotent in a similar way as the induced pluripotent stem cell, which I will talk about later. So embryonic stem cells are pluripotent and not totipotent because they cannot generate extra embryonic tissue such as the placenta. but they can generate other basically germ layers, including endoderm, mesoderm, and ectoderm. And these germ layers basically form during gastrulations. So the cells from these germ layers are then called multipotent, and they can then basically specialize in different unipotent stem cells that can regenerate in the same tissue, such as dermatocytes. So embryonic stem cells derive from this inner cell mass of the blastocyst, and normally the blastocyst, by blastocyst we mean the stage of pre-implantation of the embryo, around 4 to 7 days after fertilization. And this cell of the inner cell mass can be basically extracted, dissociated, and then propagated in culture, and then used to differentiate in different cell types. So, but there's stem cell in everyday life and in the adult tissues, as I mentioned. So we know very well the skin stem cells that are self-renewing, and they differentiate into the various lineage of the skin, and you can see it here, or down here in the epidermis layer, or their muscle satellite cells, which are typically quiescent, but upon stimulation they can activate and then proliferate to regenerate the muscle. But now talking about the induced pluripotent stem cells, these basically are derived from specialized terminally differentiated cells that undergo what's called reprogramming, and this is the expression of particular oncogenes indicated here, OCT4, CMYC, KL4, and SOX2, which have been discovered by Shinya Yamanaka, who got the Nobel Prize in 2012. So there's various ways to reprogram stem cell. I'm not going to go into too much detail, but just to give you an overview, there's viral transductions methods in which basically these factors are inserted in viral vectors, including Sendai viruses, which is an efficient way of reprogram stem cell. There's DNA-based induction methods, and these are just transfection, but they're lower efficiency, so you need a higher donor cell population. And there's other methods such as mRNA transfection or also recombinant proteins methods. And basically, the advantage of using pluripotent stem cell is that this cell can be derived from adult somatic cell, and then basically can be derived from any genetic background, and this allows the creation of a patient-specific stem cell, for example. So the reprogramming methods depends on various factors, including the donor cells, obviously, but also how the percentage of these factors, how they are present in the culture, the cell proliferation states of of the donor cell, and various other conditions such as epigenetic factors and culture conditions such as the presence of lower or higher oxygen. For example, hypoxic conditions have been shown to improve reprogramming in both mouse and human cells. Then the cell undergo these reprogramming waves, and reprogramming waves basically include the first wave in which the cell undergo transcription and chromatin changes, and these affect what's called the mesenchymal to epithelial transition. Then there is an intermediate phase of gradual changes, and these basically starts to induce the early expression of pluripotency genes. And then the second wave is basically involves the establishment and maintenance of the pluripotency. So here, basically what I've told you, that we can classify these cells based on their source. So we've mentioned embryonic stem cell that derive from this inner cell of the blastocyst. We've mentioned adult stem cells and induced pluripotent stem cells. But there's also cancer stem cells and other fetal stem cells, for example. Stem cell can be also classified based on their potency, as we mentioned, the totipotent stem cells of the zygote, which basically can give rise to an entire organism, including extraembryonic tissue. The pluripotent stem cell can give rise to the type of tissue within the embryo, and then multipotent and unipotent stem cells, which can give rise to a limited range of cells. So what are the signals that can affect stem cell specification? So we know that there's external signals, including physical contact between cells and chemical secretion by the surrounding tissue. And we also have internal signals, which are basically genetics and epigenetics and the background, essentially, of the stem cell. The other interesting thing to consider is basically the state of pre-implantation or post-implantation. So if we derive stem cell from the inner cell mass at the pre-implantation stage, we obtain a population of embryonic cell that is more mature, immature, or naive. And whether if we extract cell from the post-implantation, what's called epiblast, we basically obtain a population of primed pluripotent epistem cells. So these are more prone to differentiation, and they are called primed. These two stages basically depend on the leaf signaling pathway, and an inhibition of the leaf signaling basically promote differentiation, where the activation of leaf signaling pathway would reverse this stage. So in terms of quality controls that we need to consider when working with clinical grade human induced pluripotent stem cell, if, for example, you are planning to use this cell for regenerative medicine, we have to include several tests, and one of these tests is the short tandem repeat analysis. These ensure for basically unambiguous identification, and these short sequences can be basically used to monitor the identity of a cell type, of a cell line, to confirm the relationship between your iPS cell and your parent line, to establish a profile, or, for example, to rule out certain contamination. Then we have to also perform a microbiologist test, and the mycoplasma test, which is the most common contaminant, unfortunately, of stem cell, has to be checked regularly. Because stem cell accumulate culture-driven mutation over time, it's important to include a karyotype analysis and what's called the residual vector testing, which basically is to ensure that your iPS cells have a zero footprint, what's called a zero footprint, which means that they don't have any residual transgene sequences of the reprogramming vectors that you've used to reprogram the cell. So it's good to test for these sequences. Obviously, you want to know whether they are viable, and characterize the viability with flow cytometry is very important. Checking for epigenetic analysis and histone modification or DNA methylation. And I will go into more detail about the phenotypic assays that are important to do. So recognizing stem cells and colonies between differentiated and undifferentiated colonies is crucial to the success of your stem cell line. And basically, stem cells have a really unique and distinct morphology, with these very clear defined borders that you can see here, and you can appreciate with these images how they also expand over time, and how they sort of merge together, these colonies, to a state of more like confluent here. So here you can see the other feature is this high nuclear to cytoplasm ratio, and you can see it very clearly, this very large nuclei here. And this very defined border. So here the cell have this very, very high nuclear to cytoplasm ratio, and this dense nucleoli. Usually they have one prominent nucleoli. And this is because stem cells are in an hyperactive transcriptional state, and they are their chromatin is usually in an open conformation. So this is some of the characteristics that you need to look for when distinguishing your colonies. So the hyperactive nucleus is basically is one of the features, but the translation rate is quite low. So they have a high, they are hypertranscription. They are in an hyperactive transcriptional state, but they have low translation. And this is compared to differentiated cells where they have high translation, but they have also multiple nucleoli. So this is some of the feature you should look for in your culture. So in order to recognize differentiating colonies from non-differentiating colonies, here I put these examples, and you can see next to the other. Basically, you can very clearly see that your IPS colony here has very clear defined borders, whether this differentiating colony here appear more loosely. The cells appear more loosely located with more rough borders, and you see like there's more spaces in between cells. Whether in your pluripotent cell colony here, the cells are very tightly packed and they are flat. Here there's another examples where you can see on one side of this colony there's rough borders and the cells are starting to differentiate and escape from the colony, whether on the other side you can see this border is still very sharp and the cells are tightly packed. So if we look at the ultrastructure analysis of an embryonic stem cell cytoplasm, these are H9 that are grown on fibroblasts, mouse fibroblasts. We can see several features, including a lot of ribosomes, free ribosomes, and polyribosomes. These elongated mitochondria here that I hope you can see, and also these lipid droplets that are associated with this tiny glycogen rosettes. So this is sort of like how the cytoplasm of an embryonic stem cell look like. The other thing you can do to recognize your colonies is to perform a pluripotency markers analysis, and these are just some of the pluripotency markers that you can choose, such as NANOG, OCT4, and SOX2. You can see here in sequence, and this TRA160, and you can basically look at the expression of this marker also by Western blot. So what are the ingredients of the media that we use for culturing stem cell, and what are the key factors? So first of all, there's been several studies to develop a serum-free culture media for human pluripotent stem cells, and this is because obviously the serum is a source of variability. As well as it comes from animals. So it's better to obviously implement a system that doesn't have serum. So this is there's a study here published in Nature Methods in 2011 where they basically discover the simplest culture medium condition for growing stem cell. And these are the simple 8 ingredients here. And this media, in fact, is called E8. And basically, it's a DMEM-based medium, DMEM-F12-based medium with pH with sodium bicarbonate. And each of these ingredients has been tested separately, and they all seem to be necessary together to provide sufficient proliferation, extensive cloning efficiency and survival, as well as like long-term pluripotency. But there's also other culture media. There's several now, and there's this M-teaser, which is a more complex media, also serum-free. And this basically there's different. There's M-teaser 1 will actually contain bovine albumin, so it has some source of serum. But the M-teaser 2 is now animal protein-free, and both these media have been shown to support long-term feeder-independent culture. Survival. And then the media that we use, for example, in our lab is the StemFlex media, which is by far probably the best stem cell media so far, and has better stability over time. And you can also skip days for feeding rather than feeding every day. And this is because the StemFlex media has this stable FGF that seems to be helping basically the colonies and promote survival over time. In terms of coating, you need to consider that there's various type of coatings. Vitronectin, laminin, fibronectin all support human embryonic stem cell growth, but matrigel is one that we prefer in our lab. There's also been these studies comparing actually different coating, matrigel coating with M-teaser 1 compared to vitronectin and E8 media, and this study has shown that matrigel coating with this media is actually shown higher expression of pluripotency markers such as NanoVed or SOX2, and also a higher number of emerging colonies. So in terms of passaging, how do you passage and you keep your cells regularly in culture? So we normally like to keep our cells at not too high confluency, and so basically there's various methods for passaging the cells. There's enzymatic dissociation using collagenase, for example, manual passaging using scratchers, cell scratchers, or trypsin. However, trypsin is kind of like a bit harsh with the stem cells, so you want to use different inhibitors that promote survivals, and I will talk more about ROCK inhibitor later. So the one that we prefer is a DTA, which indirectly suppress the cell-to-cell junction via chelation of divalent cations. And this is like a more gentle way to passage cell, because stem cells are very fragile, and especially when they are in a single cell dissociated form, they tend to undergo apoptosis. So you have to be quite careful when passaging the cell, and more gentle compared to normal cell lines. So this is just an example on how we do passaging in our lab. I mean, this might be quite simple, but it basically just to show that we coat our plates in matrigel, and we incubate the plates for half an hour, 37 degrees. Then what we do is that we wash the cell in EDTA, and we incubate the cell with half a ml of EDTA if we use a 6-well plate for 5 minutes. We remove the DTA, and then we use part of the stemflex media that we use in culture to basically gently spray the well and detach the colonies and the proportion of colonies that we want to passage into the next well with fresh media. So this is just an example, but there's other methods as well. In terms of freezing and thawing, as I said, stem cells are prone to apoptosis on cell detachment. And so basically to inhibit this dissociation-mediated apoptosis, there is a range of inhibitors there to basically improve survival. And one of the most studied now and the most probably used now is this ROCK inhibitor, or this Y27632. Basically, you want to use that with both when you freeze and when you thaw your cells. So you dissociate your cells using a DTA, then you basically add this inhibitor in your cryopreserving media, which can have the MSO, obviously, and then you can aliquot and freeze your cells. When you thaw your cells again, you want to re-add your ROCK inhibitor into the culture media, and then remove it after 1 day of when the cells are in culture for 1 day. So this ROCK inhibitor has been shown to improve cell survival. You can see from this study here, and you can see where it actually acts. It basically blocks the phosphorylation of myosin, basically blocking this whole apoptotic cascade. And they've used it here to treat different freeze and thaw cell or passage cells at different days. And you can see the condition with no ROCK inhibitor has much less survival compared to the others. So in terms of feeder-free and feeder-dependent, so basically the advantage of this layer of mouse embryonic fibroblasts that grow underneath your colonies is that this layer can provide various factors, various molecules to promote the survival of your colony, and also can prevent uncontrolled proliferation. However, it's not just more laborious to have another layer of cell to work with, but it's also there's other risk. And for example, the risk of exposure to exogenous antigen and pathogens, and the variability which come with serum, because obviously these mouse embryonic fibroblasts need serum in order to survive, and the serum has, as we know, the downside of variability and more complex to work with. In terms of proof of stemness, how do you prove that your stem cells are actually pluripotent? There's various assays. for example, the protocol for differentiation, and you want to see whether your stem cell form a full embryoid body structure. And this would allow for if your stem cell are effectively pluripotent, to the formation of the different germination layer, endoderm, ectoderm, and mesoderm, and you can study that, or you can generate, for example, other structures such as organoids. There's the teratoma assay, and this basically consists in the injection of your pluripotent stem cell in an immunocompromised mouse. This will generate the formation of, will induce the formation of a teratoma or a tumor, and you can then analyze histologically the formation of this tumor to verify what your stem cell actually generated and if they produce this tumor. And then there's chimera assay, which is probably the most rigorous type of test of pluripotency, which enable to test the contribution of your pluripotent stem cell into the embryo. And you can see this like completely green embryo here. So one of the application I'll mention today is the obviously my favorite, which is the generation of organoids. And here you can see just a variety of organoids that have been established over the course of the past decades now. And you can see this beautiful skin organoid with hair follicles, or these tissue-engineered mini guts. They are generated in the luteal flap, kidney organoids, and pancreatic organoids. So there's various type of organoids that now we can generate using stem cells, and there's multiple applications. So you can generate mesendoderm-derived organoids, such as the intestinal organoids or the kidney organoids, and these basically the signaling pathway that allows this the maintenance of this mesendoderm tissue are essentially keeping TGF beta signaling high, whether in the more neuro ectoderm or ectoderm derived organoid have a lower signaling, TGF beta signaling. So we use, for example, knockout serum replacement media, and to keep this pathway low. And today I will mention the cerebral organoids protocol and what we use in the lab routinely. So in the Lancaster lab, we generate these organoids by starting from human embryonic stem cells, or also induced pluripotent stem cells. What we do is that we aggregate them in culture in combination or without these microfilaments. These microfilaments simply promote the attachment of the stem cell and the generation of what's called this neuroectoderm structure. So we form this micro pattern embryoid body, and micro pattern because they are basically patterned towards the development of this germ layer called the neuroectoderm or ectoderm derived. And then we basically add this gel called matrigel to embed the structure, and this promotes what's called this reverse polarity. And what this causes is the formation of this sort of neural tube-like structure that you can see all around here, that basically have a lumen toward the inside, and they have a apical-basal polarity. And you can see these matrigel droplets. This is how we embed our organoids, and the tiny conglomerate of cells now that are starting to organize are in here, this tiny little white dot here. And so then we can basically grow this for a very long time, and we can then section them and perform various type of analysis. So here are just some examples of images that we can get from these organoids. And if we take a cross-section of the brain and a cross-section of one of our organoids, this is obviously at very early stages of brain development. We're talking and up to the second trimester of gestation. But what this organoid can do is that they can actually recapitulate the cytoarchitecture of an embryonic brain at that stage, with as I said, the apical basal polarity and all these various ventricular, subventricular, and cortical plate, these various zones, and they actually form all the initial stem cells form actually the radial glial, and then they progressively differentiate from intermediate progenitors of neurons, and then neurons, and these neurons then basically align up here in the cortical plate. So the application of organoids are multiple, and there's been a number of studies published on this. And for example, one of the studies one was on this P10 mutant, which is basically a mutation in this gene called megalencephaly. And you can see that the organoid with this mutant are in fact they have a various increasing in this folding and in this hyperproliferation that is not present in the control. And this is interesting because we cannot model this disease in mouse models, but organoids are proven to be useful now for studying human specific diseases. We can look at differences between species and study evolution. And these are some of the images from my lab where we can actually culture gorilla stem cells and generate gorilla organoid and compare them to the mouse organoid. And we can see differences in the formation of these neuroepithelial structures. Organoids have been used to study, for example, Zika virus and study different compounds and screening of different drugs that inhibit the progression of the virus. And finally, they can be used to study some of the neurodegenerative diseases such as Alzheimer's disease or Down syndrome, because we can look at the formation of these extracellular plaques or amyloid plaques in these different conditions. So just to conclude briefly, basically what I want to say to finish this is that there's different obviously technique comparing 2D and 3D technique, and this is just up to your biological question to what technique you want to choose. And to do 2D techniques obviously have the advantage of a lack of complexity, and they're easier to use and more cost effective, whether 3D cultures and using stem cell for more complex 3D culture can have a higher biological relevance, or they can model cell to cell interaction, but are obviously more complex and more costly. So with this, I would like to thank you very much for your attention, and I'm happy to take any question.
Thank you, Laura, for that presentation. As a reminder to the audience, don't forget to submit your questions for our instructor to the Q&A tab on the right side of your screen. So we already have several questions, and we'll get to them. Now, Laura, let's talk a little more about microbiology testing, about how you maybe go about that and when it should be done throughout the life of a culture. For example, before or after cryopreservation.
Yeah, that's a good point. Actually, I think you should always store the sort of mycoplasma testing information when you're also storing your database of cell. When you add the details, for example, of your vials that are in the liquid nitrogen. For example, what we do in the lab is that we add information whether that vial was tested or not for mycoplasma. So then we know when we throw that vial that if that has been previously tested. So I think has to be done routinely, but it's up to you. It's difficult to tell exactly how frequently, but it has to be done routinely. Yeah.
Great. And do you use similar protocol and media for the human cells and also for perhaps primary mouse MSCs?
Yes, that's what we do. Basically, we culture in the lab both human stem cells and mouse stem cells. And the only addition that we have for the mouse stem cells is that we add an amount of leaf in the media, which promotes basically the mouse cell survival because they're basically primed. So you want to add the leaf in the culture, but depends on the state of your mouse embryonic stem cells. So and when they were sort of dissociated initially. But yeah, that's important to consider that. But essentially, the media is the same, apart from this leaf that we add. Yeah.
Great. And for things like karyotyping analysis or the SRT analysis, how frequently do you go about doing those? And again, maybe at what passage number would you perhaps start looking at that?
Yes. So for karyotyping is really important if you're doing sort of, if you're using iPSC for like clinical purposes. I wouldn't say you have to do it that frequently if you're just using it for other purposes, such as like testing drugs or screening. I guess it's recommended at the very beginning when you start working with your cells, and maybe after a year. And then for the residual vector testing is the same. Like you also want to make sure that you have no residual transgene in there, but that's mostly for example, replacement therapy or things like that. So yeah, I wouldn't say that you have to do that frequently, but you have to test perhaps that, yeah, before, yeah.
Hmm.
um yeah, that's, yeah, okay.
Great. Um,
Mhm.
is it important to culture stem cells in a controlled oxygen environment, and is it becoming more common to do these types of things?
Yeah, that's a very important one. So there's been studies that show that if you reduce, for example, oxygen to 5% compared to 21%, you can actually improve the reprogramming of mouse and human cells. So that's kind of like something that is becoming more and more, I guess, more and more popular now to do that, and seems to be efficient. But yeah, I guess there, that depends on also on your donor cells and other, there's other things to consider. Yeah.
Great. And you talked about kind of detecting stemness. Um, how do you go about detecting whether you've successfully reprogrammed stem cell into the particular cell type you want?
um you mean if you reprogram your stem cell into iPSC or into a specific specialized cell type?
Yeah, specific specialized cell type.
So if you want to reprogram your stem cell in, for example, um, your type of interest, then you have to, I mean, I guess you follow various protocols that are published in order to generate that specific cell type, and then you do a characterization of your cell type, including specific markers that are expressed in that cell type, I would say.
Mhm.
Yeah.
Okay, great. Um, so one comment is that when it comes to the multiple nucleoli in differentiating cells, is there any kind of gray area, or is that pretty much a rule that differentiating stem cells will have multiple nucleoli and that's what you can look for?
I mean, I wouldn't say that's the first thing you want to look for, because, yeah, I am not entirely sure how sort of established that is. But I would say that the characteristic that you want to look for is the cytoplasm, nuclear to cytoplasm ratio. How large is your nucleus compared to the cytoplasm, and the defined cell borders? Those are the main features, and it's recommended in your lab, I think, to have like some images of both, like, good colonies versus differentiating bad colonies, so people can have a look at what it is, and sort of have an idea, because it takes quite a bit of practice, I guess, to look at that. So yes, that's something that you should look for. Yeah.
Mhm. Great. Back to the assays that you went over for proof of stemness. Are there any particular pros and cons between the three different methods? And, it seems like the mouse methods might be a lot more labor intensive. Do they have particular pros over the in vitro method?
Yeah, it's true. It's definitely more labor intensive. I would say the generation of the embryoid body is the probably the simplest and most efficient way to look for that. So you can see whether your stem cell, when you aggregate them, they form the basically the three germ layers in the embryoid body, and looking for, yeah, for that formation.
Mhm. So for in your brain organoids, how are the ventricular cells getting nutrient supplements into them?
Sorry, can you repeat that in
Uh,
your?
so in the brain organoids, the
Yeah.
ventricular level cells, how are they getting nutrient supplements?
Yes, that's a very good point. I mean, we keep them on a shaker, so that after a certain time in their development, they can grow quite large. So after a certain time, we have to move them up to a shaker, which promotes oxygenation and perfusion of the media. But it does get in the cells. However, this organoid, if they reach a certain stage, they do have this necrotic core in the middle. So what we focus on is basically all the exterior part of the organoid and this sort of ventricle-like structure. But yeah, it's one of the problems with organoids is definitely that we don't have vascularization. However, there's another method that we recently published from our lab, which is basically involving sectioning of organoid with a vibratome, and then we keep this thicker section in culture alive, and they can really survive much longer because they can be exposed better to the media.
Great. The next question is regarding coding. Is coding appropriate for 3D culture, or is it better to not use any coding? Does the coding kind of affect the dimension of the cells?
What do you mean with coding? Sorry.
So coding like coating the bottom of the plate.
Coating. Sorry. Oh,
Uh-huh.
sorry.
Sorry.
Yes, coating is definitely important. Yes, stem cell are definitely need to be grown in coated plates, and that can affect their differentiation as well. I think so. It's it's important to coat stem the plate that you're using with either vitronectin or matrigel if you can.
Mhm. Is there any cell passage limit for stem cell culturing? Do they eventually just tend to differentiate or die off later on?
Yeah, that's a very good point. That's an important question, I think, in the field, and there's a lot of still confusion on this. But we tend to not keep our stem cell for too long of longer passages because they tend to accumulate mutations, and even like large chromosomal rearrangements can happen. So I would say when we are reaching like passage 50 or something, we are tend to like then discard them, and we try to keep them at lower passages when we do our experiments.
Mhm. Great. Just out of curiosity, what happens if you don't happen to remove the rock inhibitor after the first day?
I mean, the cell do acquire a different morphology when you keep the ROP inhibitor on. I think eventually, over time, the potency, the efficacy of the inhibitor sort of fades over time. But and I don't think it's too bad to leave it on for longer. But yeah, we don't like it to leave it longer because the morphology it's a bit different. They become a bit more spiky. So for the first day when you thaw your cell and you add the ROC inhibitor, the day after you look at them, they look a little bit more like fibroblast like, so more spiky, but then it's just the effect of the ROC inhibitor, so we remove it. I don't know if you keep it longer, but eventually you change the media, so it doesn't really, yeah, I don't think that's affecting them too much. Yeah.
Great. So is L-glutamine degradation in your cultures a problem, like, for example, F2FG degradation can be?
Uh, yes, because I mean it's not a problem if you use StemFlex because that has been shown to have this stable FGF. So, but if you use other media, that's why you have to change the media every day because the FGF sort of degrades over time. So I recommend if you don't want to worry too much about FGF to use the StemFlex media because that's way more stable compared to other medias.
Mhm. Great. And so for when using Matrigel, um, the does the technique you use to disperse the Matrigel on the plate affect the shape of an organoid?
That's an interesting question. So I guess it does because what we use to try to promote the best formation of this three-dimensional structure, that we use these sort of dimplet parafilm, these sort of wells in the parafilm, and then we add the organoid in the well, and then we basically add the droplet of matrigel. But there's other techniques, such as we call them like the sandwich technique, where we like just leave the organoid and we add layers of matrigel below and above, but that's sort of like it doesn't cause the same three-dimensional shape of this ventricle all around the organoid. Is a bit limited, and I guess yeah, that does influence like the way you apply the matrigel can have an effect. Yes.
Great, thank you. In your cerebral organoids, have you grown them long enough that you've progressed till the state where they start vascularization?
No, because vascularization derives from a different layer, the mesendoderm layer. So we don't have that layer in the organoid. That's why we don't have vascularization. But there's people that have added, for example, endothelial cells to the organoid system, and they have shown some kind of vascularization, and they have even implanted and grafted organoids into a mouse, and they get vascularized if they get implanted in a mouse brain, for example. So that's a limitation of the technique for sure.
Hmm. Great. What are some limitations that primed cells have that maybe naive cells don't? Are there some experiments that would require naive cells?
I guess I'm not entirely sure, but the prime cell are more prone to differentiate. So you probably have to like add certain factors such as leaf, like that's what we do for our mouse cells. And maybe there's other things to consider, but I'm not entirely sure about the differences in terms of then working with them. Yeah.
Hmm. Great. You mentioned avoiding animal products in human stem cell culture. Can you talk a little more specifically about, why that's a problem?
Can you repeat that? Sorry.
You mentioned avoiding animal products like serum in human stem cell culture. Can you talk a little more about why that's an issue?
Yeah, absolutely. Yeah. So, I mean, animal derived product can represent an issue because they are, first of all, the serum that you add is, there's different batches of serum that you buy, and they are there's a lot of variability. It is known that there's a lot of variability in serum production because it's basically producing like this mixture of proteins, and it's a highly complex mixture. So it's very variable between lots and between batches. So that can add extra variability. In addition, your stem cell can like differentiate more if there's serum in the medium. There's something that can basically induce some sort of differentiation. And also it's sort of the animal origin of the serum, especially if you want to use your stem cell for clinical purposes. You don't want to have them grown in a media or with the cell that are animal derived, because there can be antigens there from the animal or exogenous antigens that can affect then for example, they can produce an immune reaction in your then when you want to use them for regenerative therapies, they can actually be rejected. So it's something to consider when working with stem cells.
Great. Um, do you have any recommendations? Maybe not necessarily specific products or anything, but the type of plates that you'd like to use, like low attachment multi-well plates, or what do you typically use there?
No, we just use like for growing stem cell, we just use regular 6 well plates, normal attachment ones. When we make embryoid bodies to make organoids, we then use 96 well plates that have low attachment in order to generate basically a floating embryoid body. But for stem cell culture, no, there's no specific type of plate that I would recommend. I would say the normal regular 6 well plates. Yeah.
Great. You talked a bit about the FGF2 degradation. Does the same issue kind of apply with the L-glutamine?
I don't know that. I'm not sure.
Okay,
I have to be honest. Yeah, don't know.
that's okay. Um, and I think we have one more question. For using stem cells for, let's say, drug testing, um, is the field going towards 3D culture for that, or is for high throughput maybe drug testing is 2D culture perhaps simpler? What's your opinion on that?
Yes, that's a very good point. I think for drug testing and screening, we are moving towards more and more simpler 3D methods. But definitely, like definitely like still 2D methods are still much better to use for screening, in terms like, for example, these I3 neurons are used for like screening various drugs. But I have to say we are getting more into like also using organoids for screening, but it depends what type of organoids as well. Like, so we have to consider, because there are multiple type of organoids and some of them are much more simpler than others. So I guess in terms of drug screening, that's something that we are going towards that, but still we're not entirely there, I think. Yeah.
Great. Thank you for that in depth discussion, Dr. Pellegrini. Unfortunately, that's all
Thank
the time we have for today. For
you.
further discussion, the audience can feel free to reach out to the instructor directly. Her contact is on the screen. Make sure to follow the scientist on social media for updates on future technique talks. Our thanks to all who took the time to join us today, and special thanks to our instructor for today's course, Dr. Laura Pellegrini, as well as our sponsors, Sinobiological, the Baker Company, Biotechne, and Millipore Sigma. Have a great day, everyone. Goodbye.
Thank you. Bye.